different types of cell cultures. All these are from different cells or tissues upon which they tried. And they found that even the glial cells, means the nerve cells, can also be infected with this COVID-19 virus. So this is what the danger part of SARS-CoV-2. And if you look at this structure, this is the spike, okay? The other, which is the protruding part, are the spikes. And this is the picture. And this spike first interacts with the human cell, that is ACE2, angiotensin changing enzyme 2. So there are two enzymes, okay? So this could be ACE as such and ACE2. And this is the membrane-bound receptor upon which this SARS-CoV-2 first get receptor. Now, what is the cunningness of this particular virus? So many SARS-CoV viruses, they do go by this route of, reception. But this has something more intelligent. What is that? It acts on a particular enzyme can split the protein of its, this spike protein. When it splits, then the attachment with the ACE2 receptor becomes much more greater intensity it can take place. So it has another very interesting part of it. So just a background knowledge of angiotensin converting enzyme. It is the central component of renin-angiotensin system, which controls the blood pressure. And the function of ACE2 is to act as a counterbalance to ACE. And this ACE2 cleaves angiotensin 1 to 2, and the ACE2, in turn, it cleaves the carboxy terminal amino acid phenylalanine from this angiotensin 2. And here, ACE2 is the receptor. Where it is located? Very interesting. It is located mainly in the lung, but it is also in the intestine. It is also in the smooth cells. It is also found in the cerebral cortex, brainstem. So you can just imagine, the ACE2's location in the human body. So therefore, anybody who takes up or SARS-CoV-2 infecting that person, there is the immense possibility that it may not remain to the organs we are trying to prevent, that is the lungs. It might go from lungs to any other organs. So, and I told you the intelligent part of it, which is added to SARS-CoV-2, is the furin cleavage site. What is furin? Furin is a protease, okay? And this is also, it is similar to that of the subtilisin like peptidase. So peptidase means it cleaves peptide. And that it does, furin is there to process many polyproteins. So, and this furin has a site to attack in the spike protein. So this spike protein itself is engineered by the SARS-CoV-2 to have an additional advantage of this furin cleavage so that the ACE2 receptor binding points are more firmly fixed and therefore internalized much at a higher intensity and rapidity compared to any other SARS. And why this cleavage site important, I have just explained to you. It is important to note, please see the underline, the beta coronaviruses do not contain the cleavage site. But SARS-CoV-2, it has that cleavage site, S1 and S2 cleavage site. There is a specific motif upon which it acts. And so now, three important things have come to your mind: ACE2, furin, and SARS-CoV-2 spikes interaction. These three coordinates faster the viruses to enter into the epithelial cells that are expressing ACE2 receptor. So therefore, just a minute. So you have, if you look at the corona entry point, as a transmembrane, there is an AC2 receptor which serves as the main entry point. More specifically, this binding to spike S1 protein of SARS-CoV to the enzymatic domain of ACE2 on the surface of the cells. This is going to result in endocytosis and translocation of both the viruses and the enzyme into the endosomes located in the cells. And once it enters, it will be processed by a host protein, a host serine proteases, and the function starts. So if you look at the structure of this SARS-CoV-2 spike protein, you will be finding certain percentages, which is, this is RBD, means receptor binding domain. And this identity with SARS-CoV, any other viruses along with SARS-CoV-2 S protein is 76%. And if you look at the phylogeny of this, any person can see this picture and find out two great blocks in blue, blocks in green, and blocks in brown. And when you look at the blue, you'll be finding that SARS-CoV-2 is within this block of the bat ones. All the bat coronaviruses and the viruses that were sequenced during this pandemic are all down there as USA, Taiwan, Shenzhen, Northumbri, Fosan, Guangdong. These are all clubbed there. So it is coming under a same, cluster. Now, if you find this rat cov in the blue, Guanzai pangolin in a little bit more stronger red, and then the brownish one with Guangdong pangolin, you'll find a percentage of identity with SARS-CoV and bat. That's why, there is a strong contention that it has really come from bat to pangolin to human beings. And this is a classical example of tropism. And this is the lineage that I talked of. Now, if you look at the regions, which is very specific to receptor binding protein, the first line is the SARS-CoV-2, other SARS-CoV, you'll find SARS-CoV-2. And similarly, which I would like to draw your attention to the fact that, see the second line or the second row, none other than SARS-CoV-2 has a small bridge of PRRAR. And this RNS is the site for that furin cleavage. So furin cleavage site and the other part of the protein is bridged by a small 1, 2, 3 amino acids, 4 amino acids, P, R, R, A, and then the cleavage site. There is another S2 cleavage site at this point at the amino acids, position 697. And when we compare this SARS-CoV-2 and bat CoV's S protein, grossly anybody, if you don't look it minutely, you may not find any difference between the models or the protein models between SARS-CoV, SARS-CoV-2, the bat SARS-CoV. Right, now, this is very interesting. We tried to understand that where are all these ACE2 are expressing within the human body. So this is the expression picture of several organs, starting from duodenum, gallbladder, heart, kidney, liver, placenta, small intestine, testes, stomach. These are the level of expression of ACE2. So therefore, you can now easily assume that if there is a presence of ACE2, SARS-CoV can interact. And if there is an abundance of furin-like proteases, tale to shonai shohaga. And now what we did, that whether or not, this particular virus shall be moving from human to other organism or not. So we tried a phylogeny, as you said, that, our team, the whole research team is working on the phylogeny, the mutability. So we are looking across this problem very deeply in the level of nucleotide level. So if you see about the I mean the ACE2 of all the animals which are related to human, this means the human means domesticate a lot of animals. They eat many animals. You'll be finding surprisingly, that Homo sapiens and pan troglodytes, this pongo, the macaca, this monkey, all are in a same group. And we have rooted it with zebrafish. So even the zebrafish do have it. But if you look at this, you have a very strong polyphyletic, type of ACE2 evolution. But this gives us another contention that it might even go to infect our domesticated animals, and the reports are coming. So you are actually, now this gives you the entire picture of its genome. It's a big RNA, 29903 base, that is 29.9 kilo base. We don't call it a base pair because this is an RNA. And all T, you must have to think that these are all U's. And this is a huge, single RNA that is going to be transcribed, replicated. So there is a level of peculiarity there. So if you look clearly to this picture, you'll be finding that, there are the genes are separated, and how big are the nucleotides, the length of these genes are given. And you'll be finding that there are fragments that are getting, subgenomic transcriptions occur. So they manage the entire transcription business for ultimately translating and then transcribing the whole genome simultaneously. So the complexity lies here. Transcription as well as translation, that replication. And that is done by this big red part. So the most important package of its genome consists of RNA-dependent RNA polymerase machinery. And this has a brilliant machinery. I'll tell you why. And what we reveal will be very interesting for the scientists to take over. Now, this is, again, just the explanation of that. I will just skip very fast. Now, this is a hallmark paper that came out in Cell Research, the architecture of SARS-CoV-2 transcriptome. Very interesting. So if you look at the transcript nature, you'll be finding several canonical subgenomic RNAs. So RNAs, all having, it has to translate. So it has to have A at the end, and that it produces, and it starts its protein machinery, gets ready. So this will give you the schematic presentation of the genome organization, the canonical subgenomic mRNAs, and the virion structure. So see, this is ORF1A, then 1B, then S means the spike one, and some other very important proteins, E, envelope, membrane, nucleoprotein, and the last one is the untranslated region. This gives you a functional picture of all the proteins that we really have assembled. So I just skip that and come to a very important, that is the phylogenetic analysis of this 2019 SARS-CoV. You will be finding all these Wuhan strains are in a club like that of the coronavirus bat. And this is a picture of all the common coronaviruses and common cold viruses. The yellow part gives you that where our SARS-CoV-2 actually resides, in this green small leaf. And these are the point of divergences in evolution. And this gives you a clear-cut picture of the cunningness of this particular virus. Once it enters, so I talked about the spike. Now I will be speaking of some certain thing about its RDRP, which is a very noisy polymerase. That means it recruits error. And again, if you want to have a big replication of this 29903 kilobase, every time you have to have a supply of the primers. So who is going to give the primase function? So very important thing is that, this RDRP, which is also called NSP12 or non-structural protein 12, or NSP gene product 12's product is RDRP. There are two other proteins which is highly conserved in the total history of any other divergent, SARS, that is NSP7 and 8. These two enzymes, they complex with, RDRP and gives this molecule a more processivity. And that's why, they are actually recruiting lesser error than any other RNA viruses. So this is RNA-dependent RNA polymerase, the different regions of it. And you will be finding that it has, as I told you, the NSP8 and NSP7 is a very important enzyme which gives the processivity of this particular RNA polymerase. Now, what have we done? After these hundreds of genomes got published, we thought that where are these mutations happening within the nucleotide? Because if you look at the mutation in the point mutation level, there are only 4 possibilities of transition. And there are 8 possibilities of transversion. So if you have the genetic code in your mind, right, the third base is always a purine or a pyrimidine. So when you substitute a purine by another purine or a pyrimidine by another pyrimidine, so these type of mutations will be synonymous or silent mutation, because that will not result in the changes in amino acid substitution. While transversions are the most important ones which can change an amino acid, right? So what we found is that there should be, so therefore the transversions rate will be much higher because there are 8 possibilities. But interestingly, it did not happen. Transition is leading over the transversion. Why? The question must be coming into your mind. Is it because, this RNA polymerase has a selection? No, it is a random process. But when you look at the structure of what the mutants coming out, we have a biasness. Transitions are higher, transversions are lesser. Because you will never get those viruses which cannot replicate. So therefore, the entire system will never allow, or the phage ecology will never allow those quasi-species to exist, because they will fail to replicate if there are important mutations which is occurring by transversion at very important sites. So whatever the mutants that are coming out, right? So those are very, very important mutants, which we call as quasi-species. So that's what I was talking about, the transition. There are 4 different, type of transition can occur. There could be 8 different transversion. And therefore, you can expect a 1:2 ratio of transition to transversion, but we did not get it. We got transition more, transversions less. So what we did really, it's a very, very simple experiment that we did. We took certain entire genome from the viruses which emanated from China. The first one is the MN908-947. That is the standard reference strain that is being used because that's the first sequenced SARS-CoV-2. With that, we took Japan's one, USA 2, Sweden, Korea, Brazil. So we took at that period of time the ones that was doing a lot of havoc in those countries, and just made a simple blast. So 100, the blast results, when we put it ingeniously into this table, you'll be finding that amongst 100, the China one is just 100 of them. Out of that, 37 were 100% similar, not similar, identical. And there were 99% query coverage, 100%, but it never came below 99.93%. So that was actually guiding us to understand what's really happening at the nucleotide level. So when we studied these 13 pan genomes, that means first we studied 1200 genomes, saw its, the coverage, then we made it narrowed down to 13 pan genomes, means SARS-CoV, and we have plotted them in the nucleotide wise. So you will be finding in this 13 pan genome, you find these number of nucleotides and where it has happened, what point it has happened, which gene was it affected. Then we compared with 10 comparator genomes. So whatever you are understanding from this 13 pan genome, that has to be validated. So we took all the 46 Indian genomes by that time that was given that was accessible from the public database. So when we mapped all the nucleotides, if you look at this picture, you'll be surprised, the 7 means NSP7, NSP8, NSP9 did not have a single point mutation. Why? Yeah. And this is what, that's happening in the 10 SARS-CoV genomes in the comparator set. So we have found out the nature of the mutations there. With all this, we are now looking into that. Are there any mutation that has occurred in the furin cleavage site? So as I have just explained this before, and now after doing 3,600 genomes, so this is an enormous one. All the normal type of program will never do this. So we have to make a program for ourselves to find out really what are the mutations that are affecting this RS site, the 600, 685, the neighboring sites. So we found several mutations have occurred, but not all through the spike. So these are the areas of flexibility, which we'll speak in future about how dangerous the SARS-CoV will become. And when we stepped back into one of the important amino acid substitution at this, you'll be finding this CCT, right? Which CCT means CCU, which codes for has changed to H and L. That means C has converted to A and the mid one has converted to T. So therefore it is not always so. The transition cannot, give you a pertinent mutation. That means amino acid substitution. Now with all these, when we put them into a phylogenetic principle of these 13 genome, wonderfully you will be finding, see, USA strains have just been separated from all others. You have a polyphyletic way of thinking. That means South Korea and Brazil, they are near to each other. And with the Italy, they have formed a cluster. Then the Sweden is standing on this cluster, but a different branch length. Japan and Iran together. And one of the strain of China and India, they're clubbed together. And this is so because, you can think about the traders coming to Gujarat from China. Definitely, it has come via that route. And the strain has a phylogenetic closeness with the Indian strains. Now the question is why this C to U conversion was happening at a very high rate. Is it only because of the RNA polymerase? So we are looking back into the way we are disinfecting ourselves. We are disinfecting with the UV. So therefore UV is a very strong accelerator of mutation. So every time when you mutate, if you cannot kill the viruses or cleave the genes, if there is a silent 1 or 2 such spontaneous deamination of cytosine that is going to be carried out, and it will be fixed as mutation. Similarly, we also try to find out the type of the virus if you subject to heat treatment and if you sulfonate with sodium sulfite. And again, this sodium sulfite and metabosulfite, these are all standard disinfectant or preservative agent. At pH 7 and 65 degree, there is a hydrolytic deamination that can occur and that can convert this cytosol to uracil. And when it really changes to uracil, and if it takes that uracil inside, so all the C sites are very vulnerable to these type of external mutagens, which will be carried inside, and that is actually what our hypothesis is about. Similarly, there is another transition route of A to G, which you find in a very high occurrences. Why it happens? Because in many places, this formic acid is used as a tested biocide and for the cleaner formulation. And if this A to G conversion, we have hypothesized that, this hypothesis was based on Tolesa et al.'s work, that first there will be a hydrolytic deamination to form an intermediate hypoxanthine. Then there will be a second step of xanthine from hypoxanthine. And the third one is an amination of xanthine to guanine base. So this can also occur because of the indiscriminate uses of our disinfectants. I must end my lecture here. And this is the group to which many more have added. And we put up this preprint, which is available. You can see. And this is the first classical work on quasi-species, which emanated from the comparative genomics. We are now holding together towards moving into all other structure function analysis. So these are all my collaborators, along with there are more than 5 more collaborators throughout the world working on this program. And thank you for your patience and hearing of my lecture.
Thank you, sir. Now we'll take some queries from audience.
Yeah, please, I will.
So there's one. We can use antisense or palindromic sequence of the functional mRNA to neutralize the mRNA responsible for the production of capsid protein, resulting in naked genetic material of virus which can no longer affect other cells due to lack of capsid.
Now the question is, you have to find out really the possibilities of that particular therapy that you are thinking of, that whether or not, because you see this virus has several way of infecting and also going from one focus from one tissue to another different tissue. Yes, this could be one of the approaches and people should stay. But main approach must be, which I believe, after going through this, that is, you have to stop its interaction with ACE2. So this interaction stoppage will be the first one you can think of. And then you think that after entry, what can you do? You can disrupt. So we are thinking now that, this NSP7 and NSP8, which is highly conserved amongst all these mutants that we have seen. That means this is the region which is a secret and sacred region of this SARS-CoV, which it does not allow any mutation. So therefore, if you think about a molecule, which can interrupt NSP7 and NSP8 activity, I am very hopeful that you end up with a very good answer. Thank you. Next question.
Sir, I have a question.
Yeah, please.
Given the variability of the genome of SARS-CoV-2, how effective could be the vaccine in India?
It cannot be a good effective way, because the all antigen part of it is getting mutated at a very high rate, which Bhaskar has already said. Even if you get one, you have to wait for another vaccine after six years. So it is better, if you try preventing its two ways you can think of. It is if you can charge up the innate immunity of ourselves, which many people are working on it, and another is directly it is interacting like that of other retroviral drugs, right? For example, remdesivir was coming up with a heavy prospect, but somehow or the other it failed the clinical trials. But that is the type of thing you must think to prevent its replication, either replication or its entry. These two areas we need to work on. And the variability is high, and we have worked on this, that there are many regions, all structural proteins are affected, including the spike have a high degree of variability. So it can change, it can adapt. So it will be nice, if you target those two genes which are not affected by the mutation. It is not that it is not affected, it is not allowing that to exist in its population. Because that two principal protein is a very important target that the scientists should really focus on. Yeah, next one.
Yeah, Professor Chakraborty, I have one question.
Yeah.
Are you aware of any inhibitory molecules that are being thought about to disrupt the interaction between S2 receptor and the spike protein?
Yeah. There are standard ACE2 receptors. People are trying, even some doctors, when I was disseminating this in different social media, one of the doctor asked, because most of the cases, if you look, we have taken the statistics, most of the heart patients, they are not surviving from COVID. So therefore, the first and best answer is, first you stop this, ACE2 inhibitor. There are standard drugs that you can use for those high morbid,
No, actually, I'm not talking about ACE2 inhibitor. I'm talking about the viral spike
ACE2
protein
inhibitor.
inhibitor.
I'm talking about the ACE2 inhibitor.
No, I'm talking about, is there any search for inhibitors of the virus spike protein which will cause disruption of the interaction between the receptor and the spike protein?
Yeah. People are trying that. I will not share it right at this moment, because one of our team members have really thought of it, and that will definitely work wonder. Because some of these, residues, if it is changed by some chemicals, immediately the whole spike protein, that gets disorganized. So that is a very promising area. Now we have to test this real time. Now all the modeling part is finished. Now we need to check that. But so therefore, there are many intelligent and smart people throughout the world is thinking to stop its interaction.
Thank you.
Thank you.
Sir, I have a question to Dr.
Yeah.
Chakraborty. Will SNPs in the S1, S2 cleavage site or in the 4-nucleotide bridge that you talked about before the S1 causing mutations can render the RNA ineffective? Or are there any reports of non-infective forms due to any SNPs?
Yeah, it's a great question you have asked. That's what the answer we should look into. Once we have all these, the variation or amino acid substitution in nature, you have to test its infectivity. And now we are trying to correlate that those mutations which I have shown, we need to go back to the metadata. This is a very big data that is getting generated, and we need to see that whether those quasi species from which patient that has come, whether it expired or it revived, and how often this particular quasi species is getting sequenced day after another. So with that population genetics of that RNA virus, we can give you a definitive answer. But you can experiment, because we are trying that. The experiment is on that, whether or not it will enhance the ability to to bind with ACE2 better with that particular bridge part, or it will smoother, or whether it will give an advantage to move to some other part of it. It may not be respiratory epithelial ACE2, it can move to some other. So that's the part, lesser we speak, that better it is. Still we experimented.
রণজিৎ দা
বলুন।
Can we now bank on vaccine, or can we? How feasible is a vaccine?
People are really trying hard to vaccinate. Even the first effective vaccine that will be generated shall give an access to several crores of people for initial few days. So let that happen first. People are trying for this. Some of the regions which may not alter the antigenicity, that is people taking care of. So vaccine is a proposition, and vaccine is a very open feasibility. So people must try that, because that will give instantaneously a great relief to the suffering population throughout the world. But the problem is, because the problem that we faced with influenza, the same type, but interestingly, the influenza is a segmented negative. This is a totally unsegmented genome. The mechanism, more we understand the mechanism, more we get it, because this is completely new one, and you need to understand the biology first and then do it. But doing this vaccine part of it, people will try randomly. One vaccine of works means it's a good one. So you forget about the biology, but get the product first.
So there's
am I audible?
Yeah.
Yes,
Dr. Vomik?
Yes.
Yeah, please.
First of all, I would like to thank you, offer my thanks for the very, very good lecture, which is technical and research you are doing, all that has been reflected. And now where you are teaching in Northbound University, and I had my master's degree from that university.
You are my alumni too.
Yeah, in 1980. Now, as a layman in microbiology, I will ask one question, and whether there is any possibility that the virus may mute itself in time so that it becomes less virulent to people.
Yeah, that happens, because you see, if you look at its history, or if it has originated from bat, the bat is an organism, okay, which is hosting lot of viruses, because it is not allowing the viruses to overpopulate in their body. So it may happen if we have our herd immunity developed. Innate immunity, one of the important area we must be thinking. সবকিছুর আমি কিন্তু পণ্ডিত নই স্যার, সুতরাং I may not give you the right proper answer. এই ধরনের মানে ফেনোমেনন যেখানে গোটা বিশ্বের সবাই ব্যস্ত হয়ে পড়েছে এটাকে খুঁজে বার করার কিছু, সেখানে আপনার যেটা প্রশ্ন সেটা আমি যদি বলি অ্যাজ এ মাইক্রোবায়োলজিস্ট, ইয়েস देयर इज अ পসিবিলিটি। আরে ওকেও তো বাঁচতে হবে, ফাজেরও তো একটা নিজস্ব ইকোলজি আছে। ভেরি ইন্টারেস্টিং কোয়েশ্চেন ইউ হ্যাভ আস্কড। দেখুন আমাদের সিলেবাস গুলো যদি আপনি দেখেন, বায়োলজি সিলেবাস গুলো থেকে ফ্রম স্কুল টু কলেজ টু ইউনিভার্সিটি কোন জায়গায় কোন এমফাসিস আছে ইকোলজি সম্পর্কে যেটা আপনি চোখে দেখেন না? মাইক্রোবিয়াল ইকোলজির কতটুকু ইম্পর্টেন্স দিয়েছে, না দিয়েছে সোসাইটি, না দিয়েছে সায়েন্টিস্টরা। ফলে কি হচ্ছে, আজকে এটার পুরো ডিভিডেন্ড আমাকে দিতে হচ্ছে। Why? Because we don't understand the virus ecology. Viral ecology is an important factor. এই যে ইকোলজির যে ভদ্রমহিলা বললেন, she is to some extent correct. আমি তো ইন্টারভিন করে ফেলেছি সমস্ত ইকোসিস্টেম। ওর যে একটা বিরাট হোস্ট ছিল ব্যাটকে, হাজার হাজার ব্যাট খেয়ে দিয়েছি, ওর হ্যাবিটেট ডিস্ট্রয় করে দিয়েছি। তো ভাইরাস তো বাঁচতে চাইবে, সবচেয়ে বেশি দেখছি যে এখানে পপুলেশনটা কি হিউম্যান, the best vulnerable population where it can stay. So philosophically speaking, এই ভাইরাসকেও বাঁচতে হবে। ওরা হিউম্যান বিংস কে এখন বেছে নিয়েছে বিকজ দিস ইজ ভালনারেবল। আর যেহেতু আমাদের কাজকর্ম সব সিডেন্টারি হয়ে গেছে, করে কোনো এক্সারসাইজ নেই, নেই। ব্যাটের কেন করে না? ব্যাট প্রচন্ড ফ্লাইট নেয়। এই ফ্লাইটটাতে কি হয়, এই যে ইনফ্লামেটরি অ্যাকশনটা অনেক কম থাকে, যার জন্য আমরা একটা কথা বলেছিলাম প্রথমের দিকে, আমি কিছু সোশ্যাল ইয়েতেও বলবার চেষ্টা করেছিলাম যে হাইড্রোকুইনন, এই হাইড্রক্সিকুইনোলিন কুড অ্যাক্ট অ্যাজ আ ড্রাগ উইথ রেমডেসিভির। কারণ হচ্ছে যখনই একটা অ্যান্টিভাইরাল ট্রিটমেন্ট করবেন, ভাইরাস চলে যাওয়ার পরে শরীরের নিজের ভিতরে কত ক্রিয়া বাড়তে হবে, সেটাকে কমানোর জন্য ওই হাইড্রক্সি ক্লোরোকুইনের দরকার, যে ইমিউন সিস্টেমটাকে একটু ইনফ্লামেটরি ওই যে সাইটোকাইন স্টর্মটাকে দমিয়ে রাখে। এবং যারা এইচআইভি পেশেন্ট তারা বেশিরভাগ মরে হচ্ছে সাইটোকাইন স্টর্মে, তখন এদের এটা দিতে হয়। তো দেয়ারফর এই আমাদের পুরো লাইফস্টাইল, আমাদের দর্শন, ইকোলজিকে ইগনোর করার দর্শন। ক্লাসের সবচাইতে মাটো ছেলেটা ইকোলজির অ্যান্সার দেয়, দিস ইজ আনফরচুনেট। আর ওই যে সাপ ব্যাঙের খেলাটা আপনারা ট্রায়াঙ্গেল করে দেখান, এর যে কত বড় গুরুত্ব সেটা আজকে প্রকাশ পাচ্ছে। যারা ছোট্ট ভাইরাস সমস্ত জায়গাটায় দখল করে ধ্বংস করে, they have shown us, look back man and see what you destruction that you have done to the ecology. Habitat destruction করেছে মানুষের এই হাতের দুটো বুড়ো আঙ্গুল মারাত্মক জিনিস, it can change the habitat, এবং সেই হ্যাবিটেট ডিস্ট্রাকশনের ফল আপনাকে ভুগতে হবে। অল রাষ্ট্রের এখন একটা নীতি হওয়া উচিত যে সমস্ত উন্নয়ন কোন অবস্থাতে ইকোলজি কে ব্যতি রেখে হবে। You have to make all these organisms sustainable. এটুকু বলতে পারি।
We exploited the nature and nature is taking revenge on us. এটা তো বলতে পারি আমরা। We exploited the nature and the nature is taking revenge on us. Live and let live.
Live and let live. That should be the philosophy.
তবুও তো আশ্বস্ত হলাম যে মিউটেট করে যদি কিছুটা ভ্যাকসিন বাড়ার আগে যদি আমরা কিছুটা অন্তপক্ষে আশ্বস্ত হলাম আপনাকে শুনে যে হ্যাঁ হতেও পারে,
হতে পারে। There is a big possibility আমি আপনাকে বলছি, কারণ হচ্ছে এত এফর্ট সারা বিশ্বব্যাপী হচ্ছে। I have a strong contention, I am a very positivist thinking যে নিশ্চয়ই কোনো না কোনো ভাবে এটা থট করার জায়গাটায় অনেক এগিয়ে গেছে। কারণ রেমডেসিভির ফেলিওরটা আমার নিজের ফেলিওর। মানে রেমডেসিভির যখন এই বেস অ্যানালগটা আবিষ্কার হয়েছিল, এটা ভেবেছিল রেপ্লিকেশনকে বন্ধ, সত্যি বন্ধ করে দিচ্ছিল। পুরো সেল কালচারে সমস্ত এক্সপেরিমেন্টের ডেটা আমি দেখেছি, ইটস ফ্যাসিনেটিং। প্রসেসিং অ্যাক্টিভিটিও বন্ধ করে দিয়েছে। ফলে ওটা একটা খুব প্রমিসিং ড্রাগ ছিল। সামহাউ ইট ডিড নট ওয়ার্ক ইন ইউনাইটেড স্টেটস পপুলেশন, কিন্তু চাইনিজরা বলছে যে কাজ করছে। সো লেট আস ওয়েট ফর অ্যানাদার গুড ড্রাগ টু কাম আউট। Drug is one of the great viability.
Thank you very much, sir.
Thank you, Vomik sir. তাহলে আমি কি অফ হয়ে যেতে পারি?
Sir, yes. Any other queries?
Now, let me ask all of the participants, how did you enjoy this talk? Means this was, I was trying to give a bird view, যে সবকিছুকে ধরে কোন কোন ইম্পর্টেন্ট জায়গা।
Sir, you can just see the chat box and gauge for yourself the reactions that they are giving.
Okay. Thank you.
So, fabulous to informative. They are thanking the departments like anything for your lecture.
Thank you.
Okay, now we come to the concluding session.
Okay.
Bro,
তুমি
তিনজনকেই থ্যাঙ্কস দিয়ে দাও কলেজের তরফ থেকে। আমি তো ওয়েলকামটা দিয়েছি, তুমি থ্যাঙ্কসটা দিয়ে দাও IQAC তরফ থেকে সবাইকে, আমাদের যারা আজকে এখানে এসেছিলেন। ঠিক আছে?
Okay, so this is the last we've come to the part without which no session ends. There's a vote of thanks. First and foremost, our esteemed speakers who've kept us engaged for the last almost 3 hours. Thank you, sir and madam. It is needless to say that this webinar and indeed the other webinars would have been possible without the enthusiasm of our principal sir. He's there with us throughout all the webinars in spite of a busy schedule. Another person we need to mention is our president of our governing body, Dr. Nancy Halder, for his constant support in all our endeavors. We received excellent cooperation from all members of the governing body of the college also. It is teachers of the Department of Microbiology who came forward with the proposal of the seminar, which was taken forward by the IQAC. We special mention to Sheikh Tawsi Ahmed, a research scholar of our Department of Microbiology, who has worked tirelessly to stream live the seminar. And last but not the least, the enterprise and stimulating participation of the speakers and you participants who made this whole program worthwhile. Thank you. Another thing before we end, you'll be sent a feedback form on your mail ID. Please fill it up. This will help us to gauge our effort. Thank you. So, should we end, Progha?
Yes, yes.
Okay.